Hi, my name is Alex Greninger, and I'm an associate professor at the Department of Laboratory Medicine and Pathology at the University of Washington in the Clinical Virology Lab. I'm doing an ASM brief on antiviral resistance testing. This one's going to focus on CMV. So for CMV antivirals, we have therapeutic antivirals targeting the UL97 kinase and UL54 DNA polymerase. They're activated by the kinase or target the kinase, and then are integrated, many are integrated via the UL54, act directly on the DNA polymerase. And now we also have prophylactic drugs like letermovir that acts directly on the UL5689 terminase. So these are the genes that we're mostly interested in for genotypic antiviral testing. So you can see here, like acyclovir, commonly used antiviral for CMV, getting phosphorylated by the UL97 kinase, and then integrated via the 54 polymerase. Foscarnet directly acting against the UL54 polymerase. Maribavir working directly against the UL97 kinase. These are the main targets. And then letermovir over here with the UL5689, but mostly the mutations are in 56 for the terminase that cuts sort of the tail off as the viral genome gets incorporated into the capsid. So there's been a lot of work. This is a very, these are large loci. So increasingly you're seeing some more next generation sequencing tests being run for laboratory workflow because there's such large loci. And so the Sanger cost of getting 2x coverage required by CAP can be high. But you also see a lot of different mutations that can lead to or associated with resistance. And this is some maps here from Soonwin Chow at Oregon Health, OHSU, who's done an incredible amount of work looking both at mutations that are selected in cell culture, as well as those that are detected in clinical specimens. And he does the hard work of doing marker transfer to figure out the exact levels of resistance associated with them in sort of a more scientific manner. Here we're looking at the UL97 kinase. And you can see here in the N-terminus some of these mutations that are associated with maribavir, which is a specific locus that you have to sequence if you're looking for maribavir-associated mutations based on its mechanism of action. But most of the other ones are down sort of in the classic sort of kinase areas for the activation of ganciclovir. These are all mutations that have been detected in clinical specimens and associated with drug resistance. If you look at UL54, you have mutations throughout the exonuclease, as well as the major domains associated with the polymerase, so the sort of the palm, fingers, and thumb region that have been associated with resistance to foscarnet, acyclovir, stofovir, all these different different drugs for treatment of CMV. And then increasingly now we're seeing more use of letermovir from a prophylactic standpoint. Now, we don't do a ton of resistance testing because letermovir is a prophylactic. So if you see breakthrough in the context of letermovir, you don't want to be treating with letermovir. You want to be treating with one of the other therapeutics. So most of the time, we don't actually see a lot of resistance testing. We just transition to the drugs. There's no reason to send the resistance. But nonetheless, these tests are available. The locus is covered. And I think what's deserves special mention here is this 325 locus of cysteine to phenylalanine or tyrosine or tryptophan or arginine that basically leads to this 3,000-fold change in EC50. It's the major mutation for letermovir resistance in a prophylactic. But there have been a number of papers that come out. Seems to be as long as you don't have a lot of CMV when you're starting to treat or doing prophylaxis, that there's low amounts. There's not a lot of resistance that's coming up to this particular drug, and things it's really quite a sea change in the CMV prophylaxis and antiviral area. And that's all I have to say about CMV resistance at this time. Thanks a lot for listening, and I'm happy to take any questions offline.